Hello everyone, welcome to another video of Knowledge of Life. In the previous video, we have discussed about the basic concepts of enzyme, that is what is an enzyme, what is an catalyst, what it does to a human body or a living system, and how many types of enzymes are present and how their nomenclature are done. Now, today in this video, we will discuss about the structure of an enzyme and how does its function. So now let's start. Now, if we look at the structure of an enzyme has got a active site which helps to bind the substrate with it and form the enzyme substrate complex. So, the active site are made up of some amino acid residues which in normal state are located at a distance from each other, but while the substrate comes closer to an enzyme, the amino acid residues gets closer to each other by means of protein folding of secondary and tertiary structure, and they form the enzyme substrate complex. Now, inside the cleft of the active site, there are specific amino acid residues which binds to specific group of the substrate to form the enzyme substrate complex, and thus they begin their catalytic actions. Now, a change in the tertiary or quaternary structure of the enzyme may alter the three-dimensional form of its active site as to enhance or to decrease the substrate binding and the catalytic activities. So now, if we look at the properties of the active site, we can say that the active site binds the substrate with it to form the enzyme substrate complex, and the catalytic groups are responsible for stabilizing and lowering the energy of the transition state, which we will be discussing later. And the active site brings the conformational change of the three-dimensional structure of the protein, and it does non-covalent interaction with substrate and creates a microenvironment helping to prevent the unwanted reactions. Now, the enzyme only alters the rate of the reaction, but it doesn't affect the equilibrium of the reaction. So, equilibrium is the state when there is no net change in the concentration of the reactant or product. So now let's look at an example and let's explain this thing. Now, if we look at these two figures, then here we can see that there is a reaction where it is uncatalyzed or there is no presence of enzyme, and the second one is for the reaction where the catalyzation takes place, that is there is presence of a enzyme. So each of them have got two ground state, that is ground state when the reaction start and the ground state when there is formation of product. Now, if we difference the value of the ground state of the product with that of the reactant, then we get the free energy change or the del G value. Now, as we can see that in both of the reactions, that is in case of a catalyzed and a uncatalyzed reaction, both the del G values are same. So it doesn't alters the equilibrium of the reaction. Now, if we difference the value of the ground state of the product and that with the ground state of the reactant, then if the value is negative, that is if the del G value is negative, then the reaction is exergonic in nature and it will liberate some sort of free energy and the reaction will go on simultaneously. Now, what exactly the enzyme does to increase the rate of the reaction, that is it alters the rate of the reaction by means of decrease in the activation energy, that is the del G2 plus value. As we can see here, in case of the uncatalyzed reaction, the del G2 plus value is much higher, that is this is the activation energy of the uncatalyzed reaction, but in case of the catalyzed reaction the del G2 plus value is much lower. So the enzyme decrease the activation energy of the reaction at and it increases the rate of the reaction, as a result of which the equilibrium of the reaction is maintained, that is the concentration of the product and the reactant remains same, but it increases the rate of the reaction. Now we will explain what is an activation energy and what is a transition state. So let's begin. Now, according to the kinetic theory of reaction, a reactant molecule must collide with or approach each other with a minimum kinetic energy to form or change bonds. And at the top of the energy hill is a point at which decay to the reactant or product, that is the S or P state, is equally probable. That is, the reaction can either produce a reactant or a product in this transition state, that is this part of the reaction. Now, the transition state is not a chemical species with any significant stability, and it should not be confused with a reaction intermediate such as the enzyme substrate or the enzyme product complex. It is simply a moment in which events such as bond breakage and bond formation and charge development have been proceeded to the precise point at which decay to either substrate or product is equally likely. That is, it can either form a product or it can go back to a reactant. And the difference between the energy levels of the ground state and the transition state is known as the activation energy, that is the del G2 plus. That is the difference between the ground state and the transition state. Now, in case of a enzyme catalyzed reaction, the enzyme does a weak non-covalent interaction with the product and decreases the activation energy of the reaction. That is, it brings a stabilization of the transition state, as a result of which there is decrease in the activation energy of the reaction and it increases the rate of the reaction, but the equilibrium is maintained as there is no change in concentration of the reactant and the product. Now, the weak interactions between the enzyme and substrate are optimized in the transition state. Now, how does it took place exactly? Let's explain this with help of an example. Let's take an imaginary enzyme, that is stickase, which is designed to catalyze the breakage of a metal stick. Now, in the first case, when there is no enzyme present, the substrate, that is the metal stick, will have to undergo through the transition state, that is the bent stick form, and then there will be the formation of product, that is the broken stick form. Now, when the substrate, that is the metal stick, undergoes through the transition state, there is increase in the activation energy, and after that, when the product is formed, there is decrease in the energy level and it reaches the ground state energy and the product is formed. Now, in the second case, a stickase with a magnet line pocket complementary in structure to the stick, that is the substrate, stabilizes the substrate. Now, the bending is inhibited by the magnetic attraction between the stick and stickase, as a result of which the substrate cannot undergo through the transition state easily, and now in the end of the reaction a few product is formed, but in this case also there is an increase in the activation energy. Now, in the third one, the enzyme is complementary to the transition state. That is, at first there is a weak interaction between the enzyme and substrate while formation of the enzyme substrate complex, and after that, when the substrate undergoes through the transition state, the enzyme is complementary to the transition state and helps in breakage of the product, and it stabilizes the transition state of the reaction and decreases the activation energy of the reaction, as a result of which there is increase in rate of the reaction, but the equilibrium is maintained. Now, coming to binding energy. The binding energy is a major source of free energy used by enzymes to lower the activation energies of the reactions. The interaction between the substrate and enzyme in the complex is mediated by the same forces that stabilize protein structure, including hydrogen bond and hydrophobic and ionic interactions. Now, the formation of each weak interaction in the enzyme substrate complex is accompanied by a release of small amount of energy, of free energy, that stabilizes the interaction, and the energy derived from enzyme substrate interaction is known as the binding energy, that is the del Gb. It is the del Gb. Now, if we look at the role of the binding energy, it actually reduces the entropy, that is the freedom of motion of the reactant or the substrate, and the weak bond formation dissolves substrate and bring reactants together, as a result of which it destabilizes the ground state and stabilizes the transition state. Now at the end, if we look at the enzymatic models of action present, there are basically two types of models present. First one is the Fisher's template or lock and key model, and second one is the Coastland's induced fit model. At first, let's look at the lock and key model. Now, according to this model, the active site of the enzyme exists in the proper conformation for catalytic activity even in the absence of substrate, and the active site binds to the substrate without any change in former's three-dimensional structure, and the substrate fitting into the active site is just as a key fits to the lock. Now, the model can explain the specificity of many enzymes for only one stereoisomer of the substrate, and it can also explain the sequential binding of the coenzyme or the cofactor and the substrate to the enzyme. But it cannot explain the change in enzyme activity in presence of ligands or the allosteric modulators which bind to the enzyme at a site other than its substrate binding site. Now, coming to the induced fit model, it is a much accepted form of model than that of the Fisher's template or lock and key model. According to this model, the three-dimensional form of the active site possesses a certain degree of flexibility and doesn't suffer from the rigidity of a fixed template. It proposes that the active site doesn't have its fully active and complementary conformation in the absence of the substrate, but either on the approach of the substrate, the flexible active site changes its conformation to the final catalytic form with a tighter binding to the substrate. And as a result, such a flexible active site can explain complex saturation kinetics, competitive inhibitions, and allosteric modulations. So, it is a much accepted form of enzymatic models of action. So hope this video will help you, and if you have any kind of query regarding the video, you can put a question in the comment box, and if you like the video, please subscribe our channel and share it with your friends. Thank you.